Hello, my name is Chiara and I'm a postdoc at the University of Innsbruck in Austria. When the Berlin Wall fell in November 1989, I was only a future probability. I was born over 5 years later, but luckily in a slightly less divided world. When I was a child, I actually wanted to become a pilot because I loved traveling and learning new things about cultures and people. Now I've actually taken a more scientific approach to learning more about the human nature, but I still get to travel and meet fascinating people all over the world, even in a lockdown like I'm doing now. So in that sense, my wish has actually been exceeded. My research is breaking the wall to personalized preventive medicine. Preventive medicine is about identifying risk factors and acting before disease strikes. You've probably all seen this. Blood pressure monitors are an excellent tool to track risk for cardiovascular diseases such as heart attacks. If you have high blood pressure, you can do something about it. Consume less salt, be more active, take medication. And using the same tool, you can check whether your preventive action was effective. This is personalized preventive medicine in cardiovascular diseases, and it has drastically reduced mortality over the past 3 decades. However, I work in cancer research and we don't have such a tool to predict and monitor individual cancer risk over time yet. This is our wall, and our research group is trying to break it. Cancer development is complex and there's no one good risk prediction factor for it. Cancers are caused by complex interplay of genetics, lifestyle factors, and the environment. And importantly, there's a high individual component to it as well. For example, not everyone who smokes will get cancer, but then people who stop smoking may still get cancer. So how do we predict risk in such a complex scenario? Enter the stage, DNA methylation. It is an extra layer of information superimposed upon our DNA. If you imagine DNA as the hardware of a computer, DNA methylation is a software telling your computer what program to run. This program can be changed by your environment, for example, by smoking, what you eat, and so on. Cancer cells have a very distinct methylation pattern. Excitingly, we and others have found that this pattern can exist years before diagnosis. Using non-invasive samples such as cheek swabs, we could now predict the individual risk for several cancer types using DNA methylation. To finally knock down the wall and start monitoring individual cancer risk over time, we will soon be starting a new study. We will help healthy individuals with increased risk, such as due to smoking or high body mass index, to decrease their risk by stopping smoking, intermittent fasting, or increased exercise. And we will look at whether we can see a decrease in cancer risk based on DNA methylation. To summarize, DNA methylation-based risk prediction, combined with simple preventive measures, may usher in a new era of personalized preventive medicine. Cancer risk prediction has previously only been able to achieve poor accuracy because researchers were either using only genetic or only lifestyle factors. But our approach combines both, and we have an objective, quantifiable signature. Importantly, we can actually predict the risk for several cancers using only a single non-invasive sample. By improving individual risk prediction and implementing simple preventive measures such as more frequent screening, being more active, or stopping smoking, we will drastically reduce the incidence of cancers and ultimately not only save lives, but also improve the quality of these lives and contribute to a healthier society. It's actually not yet clear how effective preventive measures are on an individual level, but this is what I'm going to be looking into with my new study. I wish this was more poetic, but actually most frequently it is bugs in the code. Science is hard work, and often it is just incremental improvements in troubleshooting.